So in the development of a vaccine against the HIV, which is the main obstacle? Because after so many years, we don't have still an available one. So it's the biology problem, it's a clinical problem. What do you think?
So I think there are three major biological problems. One is, and that would be true whether you're talking about a prophylactic or a therapeutic vaccine. So one problem is the envelope protein that sits on the outside of the virus is the most heavily glycosylated protein known in nature, I believe. So that makes it very hard to have a specific antigenic structure that can be recognized by neutralizing antibodies, which would underlie any successful, would be a key component of any successful vaccine. So that's one problem. So it's very hard to make neutralizing antibodies, although some do exist. The second problem is the huge sequence variation of the virus itself. So different strains of virus can differ in about 30% of their sequence. And when you consider that flu is a much less variable virus, and even there we only have seasonal vaccines, so to make an HIV vaccine that's spread across all the different variants would be tremendously difficult. And the third, I think, is the issue of actually getting rid of the virus because of its rather unique lifestyle in that it integrates its DNA form of its genetic information into the chromosomal DNA of an infected cell. So there's no way of eradicating that infection without actually killing that cell. And so that's a feature of all retroviral infections, in that they are lifelong persistent infections. So I think those are three huge barriers or problems that have to be dealt with any successful vaccine. Now that is not to say that progress isn't being made. For example, the trial in Thailand, the RV144, I think it was called, in Thailand, that was completed about 3 or 4 years ago. There was certainly some evidence for a positive effect of vaccine versus placebo in terms of transmission rates. Although it is true, I don't believe that disease progression is any different once you are infected in that particular trial. So there is some hope there.
I know you have, or you are right now being involved in an imaging project. So could you explain us what's about? Because I think it's very innovative.
So it's a very new project funded by our Biomedical Research Centre that we have. It's a joint project between our NHS Trust partners, Hospital Trust partners, and King's College London. And our goal there is to develop probes for imaging where HIV infection is really going on in a human body. So there are a couple of ideas. One would be to take antibodies that don't need to be neutralizing, and now there are a lot of well-characterized, strongly binding HIV-specific antibodies. One idea would be to take those, label them with an isotope, infuse them into a patient, say at different stages of disease, or even when they're on therapy and have undetectable virus, to try and find out actually where is the virus replicating in real time. Because I think those are major unanswered questions. Where are the reservoirs of virus? Where are the major sites of replication during acute infection or as someone is treated? So that would be, those would be some of the questions we'd be trying to get at. But it is an interesting collaboration between clinicians, scientists like myself, as well as imaging scientists and chemists. And I think that sort of multidisciplinary approach allows people like me to tackle questions we really couldn't have thought about addressing before.
The clinical interest of projects like yours is undoubtable. But what about the funding? It's easy to obtain funds for this kind of projects?
Well, that work, as I say, is funded by our Biomedical Research Centre, which is focused all around translational research, or using humans as the experimental system. So that is a major initiative in the UK to fund work of this type. It would be hard to, I think, get that work funded in a research council right now, because it's a very ambitious, risky project, I suppose one could say. But once we get preliminary data, we would hope to be able to get to apply for funding from other sources. But as I say, our biomedical research centre does support these sort of ambitious blue skies type translational research. It's a good system to be in.
Okay, thank you very much, Dr. Malim. We wish you all the luck in your project.
Well, thank you very much. Thank you.